The overall goal of this physiologically relevant 3D culture method is to test the action of the main mammogenic hormones on mammary epithelial morphogenesis.
This method can help answer key questions in the endocrinology and mammary gland biology fields, such as how do estrogen, progesterone, and prolactin influence mammary epithelial morphogenesis. The main advantage of this standardized method is that hormone-sensitive mammary epithelial cells form 3D structures in response to hormones, in a manner similar to what is observed in vivo.
The implications of this technique extend toward breast cancer therapies, as it is a physiologically relevant culture system of the complex human breast epithelium.
We first had the idea for this method because we noticed a lack of standardized in vitro models for studying how human breast morphogenesis is influenced by hormones.
Begin by counting the number of cells in a single-cell suspension of T47D cells.
The way the cells are maintained is critical for retention of hormone-responsive phenotypes. We recommend frequent checks for estriol responsiveness in the T47D cells on the 4th passage after thawing a new vial and every 10 passages thereafter.
Then, spin down 75,000 cells per gel and use a cold pipette to gently resuspend the pellet in 1.5 milliliters of collagen per gel without bubbles. Next, use a static reducing brush to wipe the borders, top and bottom of a 12-well plate, and place the plate on top of another empty static reduced 12-well plate.